One of the things we sometimes need to do is to take a culture of microorganisms and sub it into a number of sterile broths. So we've got tubes for the different groups in the class. And so what we need to do is to take a loopful of that culture and inoculate each one of those. Now, I believe you're quite good at that, so I was going to get you to demonstrate how you would do it.
Okay, well, the method that I use, some might say is a bit of a cheating method, but I find that I have quite small hands, so when we show yours, we'll show it's a bit more difficult. So I'm going to start off just by putting the Bunsen onto the blue flame.
Yes.
Is to use. And the culture that I'm using is Bacillus subtilis.
Right.
And I'm just going to just sterilise this loop the same way that we did before. Dragging it through the flame.
Right.
Okay. It's nice and red hot for us, so let that cool. Now I'm going to just give the tube a little shake just to stir up the culture a little bit.
Why is that?
Well, Bacillus subtilis, it clumps into what they call a pellicle. It's the way that
Right.
it grows. But some teachers sometimes find that because it clumps, it's maybe not growing properly, but that's just the way that Bacillus subtilis grows.
Grows, yes, yeah.
So by just disrupting it, we'll get some of the cells just to fly off into solution, which is what we want.
Right.
So that I can now flame the lid again, the mouth of the tube. Just take a loopful out. Same as before. Put it back on. And now I want to take my sterile broth.
Yeah.
Take the lid off this one.
Yes.
And then in with my loopful of culture. Just give it a little shake around
Yeah.
there. Flame it again. Into my rack. It's a little bit of a cumbersome way of doing it. It takes a little bit longer than I'm sure the method that you're about to show us, but I find it's a bit easier and more reliable for myself.
Okay.
Okay.
Well,
so
I've got a method where I try and hold both tubes in one hand and save myself a few seconds.
of
Right.
time, sure.
So let's have a look. I've got the culture here, yes, and I've got the broth there. And what I do is I lay them along my fingers. You can actually hold them with your fingers, but it's a little bit reassuring just to put your thumb there. And I've got a bit of a gap between the two. I take the inoculating loop and I sterilise the loop so that it's nice and red. And I go for the far cap. I take it off, come back for that one, flame them, go into the culture like that, and then simply go into the sterile broth. I'll give it a little bit of shake. Everybody thinks you're busy and professional. And then what I do is flame the two, put the cap back on,
yeah.
that one back on there like that, sterilise the loop so that it gets nice and red, and I've saved myself all of a couple of seconds.
Excellent.
And I can do the next 20 for the class.